Hey, what's up and hello and welcome back to another video tutorial of Roy's Academy. In this video tutorial I'm going to discuss on the part 2 of insulin cascading or insulin mechanism that is happening within the intracellular portion or intracellular space of the hepatocyte cells. In one of the previous videos I've discussed that how the insulin hormone is cleaning off the glucose in the hyperglycemic condition and resulting in the catabolism of the glucose or anabolism of the glucose within the liver cells. So in this video I'm going to discuss that what exactly happens within the hepatocyte cell, within the cytoplasm, that is the intracellular portion, how the cascading or signaling of the insulin hormone is being partaken by other different molecules or enzymes in order to synthesize or store the glucose in the form of glycogen, in order to synthesize the glycogen and store it in the form of store it in the liver in case of emergency utilization or sudden utilization of the glucose molecules. Okay, so let's have a check that how the insulin hormone is functioning through the secondary messengers and the primary messengers as well in order to bring out the entire operation. Okay, so we have the insulin hormone. Let me represent this one as the insulin hormone. Okay, let this be the insulin hormone. Okay, now this insulin hormone is binding with its own receptor. There is a particular receptor present within the plasma membrane. This is as usually the plasma membrane of the hepatocyte cell. This is the extracellular space, okay, or extracellular matrix outside the cell, and this part is the cytoplasm or the intracellular space. And now this either end, this red colored either ends are the polar heads or the hydrophilic heads, and this part is the non-polar head. Okay, non-polar head, or the middle portion is the non-polar head or the hydrophobic. Okay, and these two parts are the hydrophilic ends or the polar ends. Now what happens, there is a particular receptor, there is a specific receptor which lies across the membrane, across this entire membrane, the trilamellar or the fluid mosaic membrane of the hepatocyte, which is called the tyrosine kinase receptor. Tyrosine kinase is actually a enzyme. Okay, so let's have a look how the tyrosine kinase receptor looks like. So let this be the head of the tyrosine kinase receptor on either side. Here also the head of the tyrosine kinase receptor is lying. Okay. And now this tyrosine kinase receptor has two subunits. Okay. Let this be the alpha. The head portion which is present outside towards the extracellular matrix is the alpha portion, and the another beta portion, the beta portion is present inside, okay, across the membrane. The beta portion is present in this way. Okay, so let us not color it so that we can easily represent this particular portion or the beta portion. Okay, let us not color it, otherwise it would be difficult to represent or write the beta symbol within this membrane, okay, within this portion. Now let this be the beta. So this one is the alpha unit, alpha, and this one is the beta. Okay, now this part is the alpha. Now these two particular subunits of the tyrosine kinase, this entire molecule here is tyrosine kinase. The tyrosine kinase is actually an enzyme and substrate of its own. So when the insulin hormone, this insulin hormone is binding this insulin hormone is binding with the tyrosine kinase receptors, the enzyme dimerizes itself. Okay, it gets activated. The particularly beta unit gets activated and dimerizes itself. Like I said, it's an enzyme and substrate of its own. That is, the enzyme is acting on its own, acting on itself and dimerizing it. Okay, the monomer units are forming double up. Okay, so this activated, now the activated portion of this tyrosine kinase, this beta subunit which gets activated here, beta subunit which is getting activated, this is nothing but activated beta subunit, the portion of the beta subunit which is getting activated, which is lying intracellularly within the cytoplasm of the membrane. This activated beta subunit will now activate another molecule which is called insulin receptor substrate molecule. Okay, now this enzyme, the tyrosine kinase, is particularly itself an enzyme which is also acting as a receptor. The alpha units are acting as a receptor of the insulin hormone, and now this alpha receptors, alpha units of the tyrosine kinase, this alpha and beta both are belonging to the tyrosine kinase. The alpha unit will help activating the beta unit of the tyrosine kinase, and now the beta unit will activate the dimeric portion, a dimerization of itself, and help creating a replicate of the beta subunit within the intracellular portion. Now this activated beta subunit would activate another molecule called insulin receptor substrate molecule, IRS1. So this insulin receptor substrate molecule again gets activated with the help of the addition of a phosphate group, because the tyrosine kinase enzyme helps in donating, it helps in throwing off the phosphate molecule, the phosphate radical to this insulin receptor substrate, which gets phosphorylated and gets activated. Now this activated insulin receptor substrate molecule will activate another enzyme which is present within the adhered within the lower portion of the plasma membrane, that is facing towards the intracellular area, that is facing towards the cytoplasm, that is particularly in this portion, in this area, the lower portion of the plasma membrane which is facing intracellularly or towards the cytoplasm, there is another enzyme which would get activated by this insulin receptor substrate molecule. The name of that particular enzyme is phosphatidyl inositol 3 kinase, okay, or phosphoinositol 3 kinase. This is the enzyme present here, phosphoinositol 3 kinase. Now this phosphoinositol 3 kinase, along with the phosphate group attached, this phosphate group would be donated to the phosphatidyl inositol 3 kinase from the insulin receptor substrate molecule. So the activated form, this particular phosphatidyl inositol 3 kinase is getting activated. Okay. This activated phosphatidyl inositol 3 kinase helps in converting another substrate molecule, helps in phosphorylating of a particular substrate molecule of phosphoglyceraldehyde or phospholipid present within this particular lower portion, lower area of the plasma membrane, which is facing towards the cytoplasm. Let's see what that molecule is particularly. So this activated phosphatidylinositol 3 kinase is an enzyme which would again help in phosphorylation of phosphoinositol bisphosphate to phosphoinositol bisphosphate to phosphoinositol triphosphate. Okay, because phosphoinositol bisphosphate in the presence of particularly in the presence of this enzyme. Okay, this enzyme will help in donating the phosphate group to the phosphatidylinositol bisphosphate. So the phosphatidylinositol 3 kinase enzyme will help donating the phosphate group to this particular molecule, which would get converted to this particular phosphatidylinositol 3 phosphate or triphosphate molecule, which is again getting activated. Now this phosphatidylinositol 3 phosphate substrate molecule, which is phosphorylated, helps in activating another messenger molecule, which is called protein, a kind of protein kinase molecule. Okay, it will help in activating a kind of protein kinase molecule, which is called protein dependent kinase. Okay, so this phosphatidylinositol 3 phosphate will help in converting the inactive form of the protein dependent kinase or PDK, inactive form of the protein dependent kinase, to the active form of the protein dependent kinase. Okay, now this protein dependent kinase, which is activated, would again help in activating of another molecule called protein kinase beta. Okay, this protein dependent kinase will help in activating another molecule. This activated protein dependent kinase will help in activating another molecule called protein kinase B, PKB. It will help in converting this inactive protein kinase B to the active form of the protein kinase B. Protein kinase B, this is activated. Okay, this molecule gets activated. Now this activated protein kinase B, this activated molecule of the protein kinase B would help in activating what? Help in activating a particular enzyme. Help in inactivating a particular enzyme. This protein kinase B, on the other hand, instead of activating, would inactivate a particular enzyme. That name of that particular enzyme is glycogen synthase. Okay, this protein kinase B would help in inactivating the enzyme called glycogen synthase kinase. Glycogen synthase kinase. This is present in its active form here. This is present in its active form. It would help in inactivating this glycogen synthase kinase. Okay, this is the inactive form of the glycogen synthase kinase. Now, why this particular step is happening in a very different manner? Okay, instead of getting activated, the glycogen synthase kinase is getting deactivated. Okay, now this glycogen synthase kinase, it needs to be deactivated. There is a particular reason why it gets deactivated. This deactivated glycogen synthase kinase would help in the conversion, would help or facilitate, would lead or trigger to the pathway of conversion of the glycogen synthase enzyme to its active form. Okay, the glycogen synthase is an enzyme. It would help in the conversion of the inactive glycogen synthase to its active form. Now let this be the glycogen synthase. Glycogen synthase. This is the inactive form of the glycogen synthase. And now this glycogen synthase molecule needs to be activated in the presence of the inactive form of glycogen synthase kinase. Glycogen synthase. This is activated form. This portion is activated form. Okay. Okay, now what happens? Why is it necessary in such a unique pathway or unlikely mechanism that this glycogen synthase kinase needs to be deactivated? Okay, this molecule particularly needs to be deactivated. Because the thing is, the glycogen synthase kinase enzyme helps in transferring the phosphate radical or the phosphate atom to this glycogen synthase enzyme. This is the enzyme glycogen synthase kinase which helps in transferring the phosphate atom to the glycogen synthase. Now, when the phosphate atom is being transferred to the glycogen synthase, it's getting phosphorylated. And what does the phosphorylated glycogen synthase do? It turns up to be inactive. So it gets inactivated. But here, the glycogen synthase inactivity is not needed because there is a particular reason why this glycogen synthase inactivity is not needed anyway for this particular purpose. The thing is, the major reason is the glycogen synthase, if it's getting inactivated, then it would not help the pathway leading to the formation of the glycogen, because glycogen synthase helps in the formation of the glycogen from the glucose. Now when the phosphate radical or the atom is getting attached with the glycogen synthase, it would not get activated, it would be inactive. Unlike the glycogen phosphorylase, which is an enzyme which gets phosphorylated, with the aid of phosphorylation it was getting activated in case of the glucagon cascading, right? We have seen. But in case of glycogen synthase, the matter or the stuffs are unfamiliar. Okay. Now what happens when the glycogen synthase, when it gets phosphorylated, it's getting inactive. And who is performing this or aiding this process? The glycogen synthase kinase. Now, when the glycogen synthase kinase, this active form is getting inactivated, so the glycogen synthase kinase cannot transfer the phosphate group more to the glycogen synthase enzyme and help thereby not aid in the inactivation of the glycogen synthase. So once this glycogen synthase kinase is inactive, this particular enzyme is totally inactive. This inactivity of the glycogen synthase kinase would lead to the activation of another enzyme which is called phosphoprotein phosphatase 1. It would lead to the activation, this would lead to the activation of another enzyme called phosphoprotein phosphatase 1. Now what is this phosphoprotein phosphatase does? It helps in removal of the phosphate group that was attached previously with the glycogen synthase anyway, or any of the phosphate groups that might be attached with the glycogen synthase molecule anyway, and thereby with the removal of this phosphate group, the glycogen synthase, inactive form of this is the inactive form of glycogen synthase. Okay, this inactive form of the glycogen synthase molecule would rather get activated. This is the active form of the glycogen synthase molecule. Okay. So, what's happening? Let's have a recap or rewind through the processes this protein kinase beta activated helps in inactivating the glycogen synthase molecule. Because if it would be the other way, that the glycogen synthase molecule, synthase kinase enzyme is getting activated, then the glycogen synthase kinase enzyme would help in transferring or donating the phosphate group to the glycogen synthase. Now when the glycogen synthase is receiving the phosphate group, it's getting inactivated. But here we don't need that here. Why? Because the glycogen synthase needs to be activated in order to perform the operative pathway of conversion of glucose to the glycogen molecule. So the glycogen synthase kinase is getting inactivated in the presence of active protein kinase B, and now inactive glycogen synthase kinase is activating or triggering the activation of phosphoprotein phosphatase 1. And now this phosphoprotein phosphatase 1 helps in activating this glycogen synthase, inactive form of the glycogen synthase, to this active form with the liberation of the phosphate, with the removal of the phosphate. So this is the glycogen synthase active form. Now what does this glycogen synthase do? Now this glycogen synthase active form, it helps in functioning, it functions on the glucose molecule. Okay, this active form of the glycogen synthase functions on the glucose molecules. Now when this active form of this glycogen synthase would act on the glucose molecule, the glucose anomer, okay, and help it in converting to the and join the glucose molecules together. It would help in joining the glucose molecules together and helping the formation of the glycogen. Okay, so this is what the chain continues like this. This is what how the chain of the glycogen molecule having the alpha 1,4 linkage continues, and this is what the entire glycogen molecule looks like. Now the glucose units, the glucose subunits or the single units of the glucose are getting converted to this glycogen molecule in the presence of this glycogen synthase, activated form of the glycogen synthase with the removal of the phosphate group, and thus the glucose is transformed into the glycogen. Now this glycogen molecule is stored within the hepatocyte cells of the liver. Okay, this glycogen molecule remains stored within the hepatocyte cells of the liver, and in time of emergency, in case of hypoglycemic condition, again the glucagon signaling is happening in order to break down this glucose to the glycogen to the glucose again, and this then liberated out of the cell. Okay, so this is how the insulin hormone is entirely functioning in order to convert the glucose molecules that are being taken out from the blood or cleaned up from the blood, and via the GLUT4 transporter it is being transferred within the hepatocyte cells of the body and then converted ultimately to the glycogen molecule. This glycogen is being stored now within the hepatocyte cells for emergency functions and needs. So this is what happens in the cascading of the insulin hormone, particularly with the hepatocyte cells, which leads to the formation of the glycogen molecule. Okay, so if you like this video, please comment down in the comment section below, and please to hit the subscribe button in order to get more such updates of latest videos on food science and bioscience as well, and also hit a thumbs up button and share the video as much as possible. Thank you.